Emily. I will be here in the background. I might turn my camera off to save on bandwidth, but I will be available for any technical problems. And as I said, you can post any questions throughout the session. Okay, thank you very much. Over to you, Gary.
Okay, thanks, Kim. Hopefully you can hear me okay.
Great, yep. Thank you.
Okay, super. Okay, so welcome everybody, and thank you for joining in this taster lecture that's actually going to be delivered by Emily. I just want to take a few moments just to say a little bit about UCL and the Department of Biochemical Engineering and what we do both in terms of research and teaching. So I think you're probably all aware UCL is one of the top global universities. Whichever ranking you look at it, it's always in the top 10, whether it's research or teaching based. And the Department of Biochemical Engineering sits within the Faculty of Engineering at UCL, which is one of the leading engineering faculties within the country, if not the world. And I'm very happy to say that biochemical engineering is let's say, a small and perfectly formed department within the Faculty of Engineering that is very much at the forefront of this discipline, biochemical engineering, which has strong links to its parents' discipline, chemical engineering. So what do we do? The activities of the department are focused in 3 areas. A couple of those are very much in the healthcare space. So we have very strong interests in things like therapeutic proteins and vaccines. So obviously you're all aware of the COVID situation at present. So we actually co-direct of something called the Future Vaccine Manufacturing Research Hub in collaboration with the University of Oxford. And that's obviously out of that has come the Chaddox vaccine that is currently helping the fight against the COVID pandemic. So the research that we do is very relevant. We also very active in the stem cell and gene therapy space. So you may have seen some recent announcements about these new gene therapies that are actually curing patients with incurable diseases. We have a very strong research program in those. And then the other area that we're involved in is an area called industrial biotechnology. And that's the sort of general area in which Emily's presentation is going to be, it covers. So industrial biotechnology is really about how we can use biological systems and processes in order to address some of the sort of big challenges that we face today as a global society. So if you think about the challenges around climate change, the challenges around sustainability, the challenges around future fuels with lower carbon emissions, then these are all within the remit of our industrial biotechnology research program. And Emily's talk is specifically focused on the use of seaweed as a sustainable resource to help us address some of these targets. So just before I hand over to Emily, just to say that from a teaching perspective, if you've applied to the department or if you have an offer from the department, then one of the UCL philosophies is very much about a research-based and a research-informed education. So a lot of the research activity we do, you will see illustrated in our talk programs. We use a lot of the latest developments as part of our practical training. And so if you join us as a taught student or as a postdoctoral student within, or postgraduate student within the department, then you will be exposed both sort of academically, but also hands-on with the types of technologies that you'll have heard about in the various taster lectures that we've presented. So after that brief introduction, I'll hand over to Emily, who's going to tell us all about the potential of seaweed as a renewable feedstock. And then I'll come back at the end and help deal with any questions and host the Q&A session after the presentation. So over to you, Emily.
Great. Thank you, Gary. Hello, everyone, and welcome. Thank you for joining today. So my name is, would help if I could move the, there we go. Has that changed for everyone now? Yep. So as Gary said, my name is Emily Kostas and I'm a BBSRC Discovery Fellow in the Department of Biochemical Engineering at UCL. And I'm going to talk to you today about some of the work that I've been doing, which, as Gary mentioned, focuses on the use of seaweed as a sustainable resource and feedstock material to make high-value products and different types of compounds and fuels that would help us build a more sustainable future. But before we get started, before I talk to you about all the exciting work that you can do with seaweed, I do have a quick question for you all to answer. And I think Kim has prepared a poll. So the question is, how many items on the screen contain seaweed, or if they use seaweed extracts in their manufacture? How many do you think either contain seaweeds or seaweed extracts in their manufacture?
I'll let Gary vote on that, by the way.
I can't vote. The window says hosts and panelists can't vote. So just to say, we're not going to test people on their responses, but it's not a trick question.
It's not, yeah, this is not a trick question. It's just to see what you guys, what you think of seaweed and what your understanding of seaweed uses are at the moment.
Let me know if you want me to share the results.
Yeah, whenever they're ready.
Okay. So I'll give you another moment to respond. Okay, I'm going to end the poll and share the results. Okay.
So that's really interesting to see that about 56% of you said all of them, that's the correct answer. So 5% said none, all but one, all but two. Well, the answer is all of them. So well done to those of you who guessed that correctly. The truth is, yes, these basic products and these simple items that we live with every day, they all contain either seaweed extracts or seaweed polysaccharides, or they might use a certain extract from seaweed during their manufacturing process. So ice creams and whipping cream and chocolate milk, these contain carrageenan, which is a polysaccharide that comes from red seaweeds, and this is used as a stabilizer in the food. Of course, jelly or jello contains agar, which has replaced the use of gelatin, so making jelly more friendly for vegans and for vegetarians. Toothpaste contains alginates, which is another type of polysaccharide found in brown seaweeds. Even dog food contains extracts or seaweed itself to help enhance the nutritional value of the dog food. And things like Gaviscon, which is an antacid, this also contains another type of polysaccharide from seaweeds called alginate. And this is important because the alginate in Gaviscon forms this sort of protective barrier at the top of your stomach, which prevents stomach acids from repeating on you and coming back up into your esophagus. So, and beer as well. Beer is an interesting one because we use carrageenan as a fining agent to clarify beer during the beer manufacturing process. So all these different items contain extracts from a seaweed, or they actually contain seaweeds in them. And this is really important because it's really important that we find alternative materials that we can use in these types of products that are more sustainable and they come from biological resources. Now, there we go. Most of you are probably aware that most materials and compounds and products that you are quite familiar with, such as fuels and plastics and different types of medicines, a lot of these materials actually contain molecules or precursor compounds that are derived from petrochemicals or fossil fuels. And of course, fossil fuel is a finite source and we're running out of fossil fuels. So unfortunately, by using these molecules to make these types of products and compounds and fuels that we're all familiar with, we're actually contributing towards global warming and climate change, because these products are not biodegradable. When we manufacture these fuels and products, we're releasing carbon dioxide into the environment, which is of course contributing towards global warming and climate change. So it's really important that we start to try and find alternative molecules and precursors that we can use to build and manufacture these types of chemicals and pharmaceuticals and medicines and plastics and even fuels that aren't derived from a petrochemical resource. So not only do we need to find sustainable molecules and precursors to make products that are more environmentally friendly, the world is we're currently going through an exponential growth phase in terms of our world global population. So as you can see here, we're currently sat about just under 8 billion people on the planet. And the UN have predicted that by the end of the century, we're just going to reach just under 11 billion people. So as well as finding new sources to make products, we also need to make products that can support this global increase in our population that is rapidly increasing at an exponential phase. So we need to create sustainable ways to basically manufacture products for the future. And we need to do this by reducing our reliance on fossil fuels, lower CO2 emissions so that we can help mitigate climate change, and move away from petrochemically derived materials by finding new sources to make tomorrow's products and also help us facilitate our increasing population. So we can do this by shifting more towards a sustainable bioeconomy and reduce our reliance on petrochemicals. And by shifting more towards a sustainable circular bioeconomy, we need to find resources or feedstocks that are highly abundant, that are more renewable, and we need to develop ways that we can manufacture products to supply our needs in an environmentally friendly way. And the technologies that we need to develop for these need to be efficient and sustainable, and they also need to be compatible with different feedstocks that we can potentially use. So seaweeds are a really fantastic opportunity for sustainability, and they offer opportunities for innovation and also nutrition. So some of you might already be familiar with some of these seaweeds. So if you've had sushi, you're probably familiar with this particular species, which is called Nori, and it comes from, it's part of the genus of Biorefining. So this is the most common one that we use for human consumption. But around the world in our oceans, there are more than 10,000 different species of seaweed. And we only really cultivate and harvest about 6 species out of those 10,000 different species for food consumption or to make products for our bioeconomy, such as extracting polysaccharides to use as a stabilizer in ice creams or to use in antacids like Gaviscon. So there are many different species of seaweed available, and they all fall within 3 types of taxonomical groups of seaweed, which depends on the color of their photosynthetic pigments. So you've got green seaweeds here. This is a famous green seaweed called Ulva luctuca. It's also known as sea lettuce. And in Asia, this is consumed quite a lot as just a healthy salad because it's full of nutrients. Here you can see kelp. These are brown seaweeds. These are beautiful kelps that you can find across our oceans, mostly in cold waters. And this particular species here has been farmed, I think, off the coast of Scotland in England. So this one here has been grown for commercial applications, but this kelp here is actually found on the seabed. So you can find these beautiful kelp forests in our oceans that grow along the seabed, and they're absolutely beautiful, and they can grow up to 10 feet in length. They're absolutely enormous. But what's interesting about seaweeds is that they are amazing carbon sinks. So just like land plants, they have the ability to photosynthesize, and they absorb carbon dioxide from the atmosphere, and they trap that carbon dioxide in their cells, and they use that carbon dioxide to generate or to make their carbon reserves so that they can survive. And I think some species of seaweed are able to uptake carbon dioxide 50 times faster than most land plants. So they're absolutely amazing carbon sinks for our planet, and they help contribute towards mitigating global warming and climate change, because they can deacidify our oceans. But as well as that, if we can sustainably cultivate these seaweeds, and we can grow them in a sustainable way to make enough biomass that will mitigate climate change, we can also use these seaweeds for applications in biorefinery type processes. And they're perfectly suitable for applications in biorefineries because they do not require land for cultivation or any additional nutrients such as land-based plants, because they grow in the ocean. They have a very large biomass yield. They have very large biomass yields and growth rates. And some of these kelps that you can see here, they can grow about 2 feet in length a day, up to 2 feet in length a day. So they're very, very fast growing organisms. And they don't really directly compete with our food sources. There's more than 10,000 different species of seaweeds, and we're only really using about 6 at the moment for human consumption. So plenty of resource available to develop these sustainable biorefineries with seaweed. But importantly, they contain very high value biocomponents that we can extract or fractionate and use these either as chemicals or products across different industrial applications, or we can use them as precursors to make other types of fuels. For example, using glucose as the monomer, as a carbon source to make bioethanol. So they're very versatile, humble feedstocks that have fantastic applications in biorefineries. But with every advantage, we do have some disadvantages with seaweed. One of the main ones being that they have a very high water content. So this would of course make transportation difficult or challenging with your seaweeds. So after you've harvested your seaweed, if you want to transfer it to a factory or to a biorefinery, you shouldn't really have your biorefinery too far away from the harvesting site, because it's difficult to transfer something that contains a lot of water and is very heavy and wet. Seaweeds also have seasonal variations in their composition. So if you want to extract carbohydrates from the seaweed, you want to make sure that you harvest your seaweeds at the most suitable time of year where your carbohydrate content is the highest. So there are these seasonal variations throughout the year that we need to consider and identify the optimal times of harvesting these seaweeds. And something that I'm going to touch on a little bit more in a bit more detail later on in the lecture is that seaweeds contain these really unique monosaccharides, including glucose, but they have other monosaccharides that are quite difficult to ferment, because not all yeast strains or not all microorganisms can ferment these unique monosaccharides found in, or sugars found in seaweeds. So finding a suitable microorganism to ferment these sugars into a fuel has been quite challenging. But I'm going to touch upon this in a bit more detail in a bit. But as complicated as this might look, we are starting to develop suitable and different types of ways and pathways to valorize and fractionate all the different parts of the seaweed and the different biochemical composition, the different biochemical components in the seaweed. And we need to do this in a way that's sustainable, so that we can maximize the value of the seaweed by recovering as many products of high value as possible in one go, or in a cascading approach, so different units or different pathways. And to do this, it's really important that we develop biotechnologies that are compatible with seaweed feedstocks, because a lot of the biotechnologies that have already been developed and are being used to biorefine lignocellulosic materials, for example, such as wheat straw, these biotechnologies are not compatible with seaweeds. And that's because of the unique biochemical composition that seaweeds have compared to lignocellulosic materials. So as well as developing susceptible biotechnologies, it's important that we also valorize as much of the biomaterial, as much of the seaweed as possible, and generate as little waste so that we could try to maximize value. But if we look a little bit more at the composition of seaweeds compared to land plants, you can see here that there's quite strikingly different. So land plants mostly contain cellulose and hemicellulose and lignin. Lignin is a complex aromatic polymer which basically offers support and rigidity and structure to the land plant and to help it stand up. Seaweeds do not contain any lignin at all because, of course, they float in the ocean, so they do not need this structural support and rigidity. And they have lower amounts of cellulose and hardly no hemicellulose in their structures, in their cell walls. But instead, they contain all these, they have a high amount and high concentrations of these other types of soluble polysaccharides. And compared to land plants, they also contain a higher content of protein, which of course is a good source and good opportunity to recover new types of proteins that we can use in the food industry from seaweeds. But if we look at in a bit more detail, in addition to the differences between seaweeds and land plants, these polysaccharides also differ between the species of seaweed and the taxonomical group of seaweeds. So brown seaweeds contain alginates and fucoidins, red seaweeds contain agars and carrageenans, and your green seaweed mostly contains olivin. And these polysaccharides are all structurally very different, and they're made up of different sugars or monosaccharides to build up these larger polysaccharides. So compared to the humble land plant, seaweeds are very diverse and very different. So this is why a lot of the biotechnologies that have used land plants to make fuels, for example, have progressed relatively quickly, because scientists have been able to break down this cellulose structure into glucose, which we can then ferment into, say, bioethanol, because glucose is always a preferred carbon source for any microorganism. But seaweed contains these wonderfully unique monosaccharides, which are not that easy to ferment into biofuels, but they offer many different opportunities for developing other types of products or other types of chemicals using these as precursor compounds. And I'm going to touch upon monosaccharide content in seaweeds in a bit later on in the lecture. So my research has essentially been focused on using one particular type of brown seaweed called Laminaria digitata, which is found in quite high abundance around the UK, and at the moment it's looking to be one of the most promising species of seaweed that we can grow in the UK. So there's great potential to build a UK seaweed bioeconomy with this particular species of seaweed. But I've been developing ways and different ways to biorefine Laminaria digitata, and I've explored different types of biotechnologies that we can use to fractionate or to break down this seaweed and recover different biocomponents from the seaweed that we can then use to make different products. So this is just an overview, a snapshot of what has been developed so far in my research, and I'm going to give you a few examples of what has been done exactly and some of the challenges that we've had to encounter. But of course, quite excitingly, what amazing products that we can make from this amazing kelp that we can find in the UK. So one of the products that many researchers have tried to make from different types of biomass in general is bioethanol. So we need to find new sources to make sustainable fuels, and of course biomass feedstocks are a very good resource and feedstock for this. But to make bioethanol, regardless of the type of biomaterial that biomass that you're using, you need to follow three main steps. So you first need to apply pre-treatment to the feedstock. And this usually involves using acids, quite harsh acids, or in some cases alkalis, to break down the cell wall structure. And for hemicellulose, for lignocellulosic materials in particular, this pre-treatment step is very important because it removes this really recalcitrant lignin component in the cell wall, and it exposes the cellulose and the hemicellulose polysaccharides. And then this makes the pre-treatment makes the polysaccharides more accessible to enzymes, which we can then use to break down these larger polysaccharides such as cellulose into the monosaccharides such as glucose, which we can then ferment into bioethanol using different types of yeast, or sometimes we can use bacteria, but we use quite often different yeast strains to produce bioethanol mostly from monosaccharides or glucose in particular. So this is the main way that we kind of have to follow these very technical steps to produce bioethanol from different types of feedstocks. And the same can be applied to seaweeds. However, because seaweeds do not contain this recalcitrant lignin fraction in their cell walls, in theory, we shouldn't really need to apply such harsh thermochemical pre-treatments to modify the cell wall structure and to break it up and expose these polysaccharides to enzymes. So one thing that was investigated was, in order to make bioethanol from seaweeds, you really need to optimize the pre-treatment conditions and try to make the pre-treatments as sustainable and as efficient as possible, so that you can liberate as much monosaccharides as possible from the material. So, apologies, somebody just walked into my office. So as I was saying, this is just an overview of how of the approach that was taken to optimize both the pre-treatment and the enzyme hydrolysis stages to liberate as much glucose as possible from seaweeds. And 3 different seaweeds were chosen to develop these processes with. One was a brown seaweed, Laminaria digitata, one was a red, and one was a green. And that's because their compositions are all different as well. So you need to optimize the pre-treatment conditions that you're going to employ on these seaweeds. So because we know that seaweeds do not contain lignin, the theory is that we don't really need to have such a harsh thermochemical pre-treatment. So very low concentrations of sulfuric acid were used as a pre-treatment. And we also trialed just water-based pre-treatments, otherwise known as autohydrolytical pre-treatments. Of course, water is very environmentally friendly, so we wanted to make sure that we could develop a process that was not hazardous, not making any hazardous chemical byproducts to the environment. So pre-treatments were a parametric study was designed where we varied concentrations of acid and compared water-based pre-treatments with different reaction times and solids loading contents. Now the solids loading is basically how much seaweed you can pre-treat in one go. So the more seaweed that you pre-treat in one go, the more economically economical your process would be. But there's always a balance, because the more material you try to pre-treat, you might encounter mass transfer limitations, and you won't get sufficient heating of your material. So this is very important. And also we did some enzyme hydrolysis optimization as well. So here we can see just a snapshot of the efficacy of the different pre-treatments that were trialed on different seaweeds, so that we can see which pre-treatments liberated the most amount of glucose that we can then ferment into bioethanol. Now there's quite a lot going on here, and I don't expect you to understand everything here, but the most important thing to realize is that each species of seaweed belonging to the different taxonomical groups required a different type of pre-treatment. So one universal pre-treatment is not suitable for each type of seaweed. So here we can see with Laminaria digitata, the grey bars here, these were the pre-treatments that contained very mild concentrations of acid. So an acid-based pre-treatment is needed for this type of species of seaweed to liberate the most glucose from it. For the red seaweed, Dilsea carnosa, it does look as though the water-based pre-treatments were superior than the acid-based pre-treatments. But as a comparison, we also investigated whether a pre-treatment was needed at all, or whether we can just directly enzymatically hydrolyze or break down the seaweed structure with enzymes without the need of a pre-treatment. And it looks as though this feedstock, this particular species doesn't need a pre-treatment, a water-based pre-treatment, because we're getting similar yields of glucose that were liberated between the non-pre-treated material and the feedstock that was pre-treated with water. So this is very interesting. So we might not need a pre-treatment at all with this particular species. But for the green seaweed, we can clearly see that water-based pre-treatments here were more preferential and better suited to this particular feedstock to liberate the highest amount of glucose. So this is important to understand, that you can't just apply the same, one universal pre-treatment does not apply across all the different types of seaweeds. We have to understand their biochemical composition and tailor pre-treatments specifically for each seaweed species belonging to different groups, to liberate as many monosaccharides as we can from the different seaweeds. Now, I'm not going to go into the detail about the other aspects that we optimized in this trial, because pre-treatment optimization and enzyme hydrolysis optimization is very time consuming, and you have to design your experiment that covers as many parameters as possible so you can find your optimum conditions. But here's just a snapshot of what we found to be the most optimum conditions for both the pre-treatment and the enzyme hydrolysis conditions for the different seaweeds. And they are quite different. So each species required a different type of pre-treatment and enzyme hydrolysis loading rate. So after we found these optimal conditions, we then generated enough liquid or enough hydrolysate, which is a liquid fraction generated from the seaweeds that contains the glucose for us to ferment. And then we did some fermentations, and you can see here that the yields of bioethanol vary depending on the species of seaweed. Now, we did not optimize the fermentation. Normally you should optimize fermentation so that you can maximize your ethanol yields. But we can see here that the yields are still relatively low, but that's understandable. But in order for bioethanol to become commercially viable on an industrial scale, you need to be generating about 4 to 5% bioethanol ABV, which is alcohol by volume, to make it viable. But these yields are still quite low, and the highest that we're almost achieving was about 1% ABV. So bioethanol production alone from seaweeds is not economically viable as a process. So we need to kind of incorporate other extractions or develop a biorefinery process that recovers other products of high value first, and then bioethanol is normally generated in a latter point down the cascade of the biorefinery. However, it's also important to make note of the fact that we only targeted glucose liberation from these pretreatments. Remember, seaweeds contain all these other monosaccharides that can also get liberated during these pretreatments, but finding a suitable microorganism or a yeast strain to ferment them is quite challenging. So one thing that we did to try and identify yeast strains that could possibly utilize these different monosaccharides other than glucose in seaweeds is we used a phenotypic microarray to basically screen many different species, different yeast strains on these different seaweed monosaccharides. So this is a very high throughput screening tool that uses the redox chemistry of the yeast of the cell, and it employs cellular respiration as a universal reporter. So essentially what you do is you make media, synthetic media that contains one of these seaweed monosaccharides as the carbon source. Then you add this tetrazolium dye to your media, and then you plate out your media onto these 96-well microarray plates. So these are very tiny volumes, about 100 microliters in volume max, which is very small. So you plate out your media onto these plates, and then you inoculate your different yeast that you want to screen onto these plates. And then what happens is, essentially, if the yeast like the carbon source, if they like these different monosaccharides, they will start to grow on these monosaccharides. And whilst they grow, they start to metabolize. And then when they metabolize, they undertake cellular respiration. And when they respire, they produce this cofactor called NADH. And when the cofactor is produced, the tetrazolium dye gets reduced by the cofactor into formazan. Now, formazan results in a color change, into a purple color change. So what this Biolog Omnilog machine can do, it's pretty cool. It can detect differences in color, and it basically uses the color intensity as an output to determine the metabolic activity of the cell. And then the machine translates this color into a number, and then we get an output, a redox intensity output number. So if you grow your strains on glucose, this is what you would expect to see after, say, 50 hours of incubating the strains on glucose. So these are your different strains down here. And because glucose is the preferred carbon source, you will get quite a lot of high redox signal intensity outputs from these different yeast strains. But when you look at a seaweed monosaccharide, a seaweed sugar, because most strains do not like, say, fucose, for example, which is from seaweed, the redox signal intensities won't be so high. However, when you start to see intensities that are relatively high, this is what you're looking for. You're looking for the odd or just a couple of strains of yeast that appear to be to have some sort of metabolic activity on these unique seaweed monosaccharides. So then we shortlist these yeast strains for further trials. So we shortlisted just 5 out of all the strains that we screened. We just found 5 that appeared to like or appeared to metabolize these seaweed monosaccharides. And we did some trial fermentations, so some anaerobic fermentations using these yeast strains. where we actually made some seaweed pre-treatment hydrolysate. So we've got some seaweed and we added some acid and we broke down the seaweed structure to liberate all these different monosaccharides into solution, which is called a hydrolysate. And we found that when we did the fermentations, we found that the majority, actually all of the yeast strains that were shortlisted, apart from one, so 4 out of the 5, were actually able to produce bioethanol, which was very interesting because now these are, of course, candidate yeast strains that require further investigation, and we can try to identify whether these yeast strains have unique metabolic pathways that can consume and utilize these wonderfully unique seaweed monosaccharides, and hopefully we can try to make more bioethanol from seaweeds. And this Saccharomyces strain was able to produce the highest amount of ethanol, about 13 grams per litre. But as I've mentioned, ethanol overall is still a low value commodity product. So in order to develop sustainable biorefinery processes, we need to also fractionate out other components of seaweed that have high value. So knowing this, we started to develop biorefinery processes using laminaria digitata that focused on the extraction of 2 polysaccharides. So these polysaccharides are quite high value and they have very interesting industrial applications. So alginate is quite a common polysaccharide that we can extract from seaweed. It's used as a food stabilizer, but it can also be used as a precursor to develop to create bioplastics. So it's highly applicable and highly versatile. And fucoidan, on the other hand, is very expensive. If you were to purchase fucoidan from a chemicals supplier, to buy just 1 gram of fucoidan, it costs just under £500. So it's a very expensive polysaccharide, and it's got these beautiful biological activities that we can take advantage of and use across many different industrial applications. So it's got antioxidant properties, it's got anti-cancer properties, antimicrobial. So fucoidan is a very highly sought after polysaccharide. So we started to develop a process based around the extraction of these polysaccharides. And then you are left with a residue fraction and a liquid fraction after you've extracted them. So after analysing this residual fraction, we noticed that there was an enriched amount of glucose in this fraction. So because cellulose is made up of glucose monomers, we predicted that the recovery of these 2 polysaccharides enriched the cellulose fraction in the material that remains in the residue. So we then developed a process that would then generate bioethanol from this residue. So we're building upon the biorefinery and kind of finding alternative places in the cascade to make bioethanol after we've extracted higher value polysaccharides. To further enhance the value of this bioprocess, we looked at the waste liquor stream. Because this, of course, most of the time this gets discarded, but after we prepared it and we prepared some extracts, we actually identified that this waste stream contained antimicrobial activities, and it was able to inhibit the growth of this bacterial pathogen called Biorefining inacipacea, which is a pathogen that is detrimental, that could cause detrimental effects to people who have cystic fibrosis. So antimicrobial activity was detected from this waste stream, and of course this is very important because this could then be used as a pharmaceutical, as an antimicrobial agent, which we need to find new sources, even from waste streams from seaweed bioprocesses, to make these antimicrobial drugs. But we also found that the waste stream also possessed antioxidant activities as well. And also the fucoidan also had antioxidant activities. So you can start to see here that when you start to build upon your biorefinery, when you're analysing waste streams that you would normally discard, it's important to try and obtain as much value as possible. So we're trying to maximise the value from waste, and it's of course very fortunate that we were able to find biological activities from within this waste liquor stream. But again, instead of producing ethanol, because ethanol is a low value product, we wanted to then see whether this residue here was suitable to apply in a pyrolysis process. So for those of you that don't know, pyrolysis is a technology, or is a method of producing 3 types of pyrolytic compact products by basically heating the biomass to very, very high temperatures, between 400 to 600 degrees, in the absence of oxygen. And when this happens, the biomass thermochemically deconstructs, it breaks down, and you get these 3 unique pyrolytic products, which of course have very good, can be used in different industries. So you get a biogas fraction that gets produced, and then you get these bio-oil vapors, which then get quenched, and you get some nice liquid bio-oil fraction. And the remaining residue, the remaining material, becomes a biochar, solid char. So the biogas you can then use, you can burn it to make energy. The solid biochar can be used either as a fertilizer, or it can be used as a nutrient agent for soils, or it could also be used to bioremediate in bioremediation. And the bio-oil fraction, this is what we were very interested in, because bio-oil, you can see here, there's this very tar-like, gloopy liquid fraction that is very, very smelly, and even to this day I can still remember the smell of this bio-oil after we pyrolyzed seaweed. This fraction contains many different compounds inside that we can use as precursors to make other chemicals or products in industry. So we wanted to basically see whether these seaweeds and the residues could be pyrolyzed by microwaves. Now, microwave pyrolysis is a very emerging technology, because it's very fast, it's very applicable, it can be used across different types of feedstocks. And we wanted to see whether microwave pyrolysis is susceptible to seaweeds and what types of products we can make with the seaweeds using microwave pyrolysis. But what's very cool about microwave heating is that it heats differently to conventional heating. So with microwave heating, you get this volumetric heating, where the inside of the material actually heats up first, gets very hot first, and then the heat dissipates out into the surrounding of the material. Whereas with conventional heating, you heat from the outside inwards. So there's this reverse heating profile that's unique to microwave heating, and this offers many opportunities because it's a different heating gradient that can result in the production of different types of products. So and also it's a lot faster. You can attain a certain temperature a lot quicker with microwave heating. So the processing times are significantly reduced. But not all we know that not all materials are good at absorbing microwave energy. So this is a very complicated graph, and I don't want you to be afraid by it, but basically what's very important to understand is that before you heat something with microwaves, you need to understand how well this material can absorb microwaves and convert that energy, the microwave energy, into heat. So we have to calculate the dielectric properties of the material, which essentially tells us how well the material can absorb microwave energy and convert it into heat. And here we can see with our seaweeds that the loss tangent, the loss tangent's dielectric profile is a non-linear function of temperature. So basically what this means, the higher the loss tangent, the more microwave absorbent the material becomes. But as you're heating the material over a temperature range, you get different profiles. It's not the same throughout. Now, pyrolysis normally takes place at these temperatures, from about 450 degrees onwards. And just before you start to pyrolyze your material, the dielectric properties, or the material, becomes very lossy, which means it can't really absorb microwaves and convert the microwave into heat. But as soon as pyrolysis begins, you get a rapid exponential increase in the loss tangent, and that's because you get the formation of biochar, and char is a very high absorbent of microwaves. So this is where essentially when pyrolysis takes place in the material. And these are fundamental characterization techniques that you have to often apply before you develop processes, different types of sustainable processes with biomaterials, because you need to apply the fundamental understanding of the characterization properties of the material to help you build your process. So just very quickly to explain to you how we did the microwave experiments and pyrolysis experiments. You can see here that we didn't actually use a kitchen microwave, which a lot of researchers actually do. We actually built a microwave rig with our biomass here. So the microwaves are generated by the generator, and they travel down into this area here where they can start to heat the material. And if any microwaves aren't being absorbed by the biomass, they get reflected back from the sliding short back to the material, and any that still can't heat the biomass go back to this Homer tuner, and this Homer tuner can calculate how much energy has been absorbed by the biomass. So this is a schematic of what happens during pyrolysis. So as soon as you start to heat your material with the energy, with electromagnetic energy, you start to pyrolyze and you char your material, and you produce these vapors which then quench into producing your bio oil. So we basically investigated different powers and times to see whether pyrolysis can take place with our residues, with our seaweeds. And what we found was that if we used lower powers for a longer time, we were able to generate a greater mass loss and higher bio oil yields. And this is what we need. You want to generate as much bio oil as possible during your process. And here you can see the effects of the different effects of the charred pellets of seaweed after we did the pyrolytic runs. But as I said, we were very interested in this bio-oil fraction because it contains many different compounds that we can use as precursors to make other types of products, including fuels, or for example, resins from wood. For wood products, we can use formaldehydes that are generated and quenched in this bio-oil fraction. But after we analysed our bio-oils, we found that our oils, because they were derived from seaweeds, contained thermal degradation products that are unique to seaweeds. And a lot of these, we found these for very interesting products that could be used as medicines. So isosorbide can be used to prevent chest pain, heart-related chest pain. Dianhydromannitol is an anti-inflammatory drug. 2-acetylfuran can be used as a precursor to make other chemicals and pharmaceuticals. So what we found was that pyrolysis is a very good technology to use with seaweeds, and we can incorporate pyrolysis within a seaweed biorefinery. So to conclude, I know I've taken up quite a lot of time. To conclude, seaweeds are fantastic to use as precursors, as renewable materials, feedstocks to use for the future, in the future landscape of biorefineries. You have to develop biorefineries that are tailored towards each type of seaweed, but overall, fundamentally, you need to apply a multidisciplinary aspect to develop these biotechnologies in order to make them truly viable on a commercial scale. And just to finish off, seaweeds are quite famous at the moment, so keep an eye out of seaweeds. They're making headlines in the news. So seaweeds have the potential to be used as a fuel and food of the future. So the work that's going on here in the department is, being echoed around in the community as well. So the next time you have sushi, just remember that the seaweed that you're eating has the potential to build a sustainable bioeconomy and future. And with that, thank you very much, and sorry for running over time.
Actually, no, that was fantastic. You're still 2 minutes before the end of the session. But so the way this is going to work is because we're coming up to close to the end. If it's okay with you, Gary, we'll go for 10 minutes at the end for Q&A, but the recording won't feature the questions, I'm afraid. We won't be able to fit that on the recording.